I'm going to go ahead and get started. Please feel free to keep those Q&A questions coming. Even if I don't answer them right away, I'll be able to see them, and so will, I think, maybe other participants. The presentation is divided into three sections, past, present, and future. So I'm just going to very briefly present a few slides just to give a little bit of context of what was the original goal of Subway 1.0 and how we're still honoring that goal and meeting it in new ways. A little bit of context. Then I'm going to quickly just review some of the new features that we've designed with and give you an actual live walkthrough. Well, you'll see me do a step-by-step analysis. I'll warn you right away, since the time is limited, I'm going to go fairly quickly through that walkthrough. But again, you can ask me as many questions as you like, and this will be recorded, and so you can go through it in more detail. And I'll point you to some other help and resources. So that's the final point, the future. We'll talk about that timeline of other features because today we're only focusing on DNA subway blue line for DNA barcoding and also talk about more opportunities for feedback. Okay, plain and simple, the DNA Subway's goal has always been to enable students and educators to do research using the same tools as bioinformaticians. So we really and genuinely think of DNA Subway not just as a platform where you can learn about bioinformatics, but a platform where you can actually do bioinformatics. It is possible to do publishable work, work that matters, work that uncovers a valid result. And that's exactly what we want in the classroom, but we've tried our hardest and will always keep trying to do is to remove as many barriers so that even if you are not a bioinformatician, which most of us educators, most of us students are not, you will have the opportunity to use those tools in a much more classroom-friendly way while achieving your end goal. DNA Subway 1.0, as shown on the screen, so familiar to several in the room, was actually launched back in 2010, and it was as a part of a project which we participated at the DNA Learning Center called the iPlant Collaborative, which is now known as Cyverse, and that was really the beginning of the project. And as much as I'm not sure what it sounds like, 2010 is actually a long time ago, certainly in web development terms. Over the course of that history, we've had more than 77,000 registered users and served nearly, I'm sure by the end of this year with Civil 2.0, as you can sort of see those numbers coming to our graphic on the right, served over 1 million sessions by the time we get done with 2026. But by our numbers, this certainly makes it one of, if not the largest bioinformatics platform designed for the classroom. So we're really proud of that capability. If we look at what has guided Subway 1.0 to become Subway 2.0, well, for Subway 1.0, we've surveyed more than 16,000 users, really getting a sense of not only who they are. In this graph, it's just focusing on the population that we really serve, which is really a heavily used in college, but also K through 12 use, yeah, also used by researchers, but really to find out from those users what features matter to them, what did they want from us in a new version, which fortunately, as you'll see, we were funded to do by the U.S. National Science Foundation, and we'll give some credits and acknowledgments towards the end. So why Subway 2.0? What did those survey data tell us? Well, there were three large categories, and I've added some details here that were motivations behind our design. The first one is to actually think about updating that design to be modern, and also what we call mobile first. So designed for the area of the area of the era of the smartphone, which was not a prevalent tool back in 2010. It was just getting its start. I think iPhone was 2008. But things that we learned were that in terms of access to technology, especially with the population of students that we were most concerned with reaching, this was funded by a program that heavily invests in preparation of community college students. We found that smartphones were even greater than laptops in terms of student usage. And so making a platform that could work well on a smartphone and designing it first for the smartphone, but then to work as equally well on a laptop is something that's really important. We also saw that some of those features, including accessibility, are important for students everywhere from students with disabilities to students in a rural environment where maybe actually the mobile device is the main source of internet connection. Um, the other major point was updating the tools and workflow options. So again, trying to maintain software from 2010 in terms of security was starting to become challenging. So things that you won't see is that we had to modernize that back end. We also have new data types. We'll talk a little bit about nanopore sequencing. And then, of course, as Cyverse transitioned from uh, a fee for service or subscription model, we also wanted to make sure that we made DNA Subway available at no cost. And the final thing in the goals of the new grant that funded DNA Subway 2.0 was to make sure that we worked to keep learning experiences aligned with new workforce demands. And I'll mention that towards the end, and there'll be some resources that you'll see in the summer. So, this is the platform. I think some indicated on the poll that they had not used DNA Subway 2.0 yet. So if you haven't seen it, well, tonight's the night. You will not see as much the fact that it works very nicely on your cell phone, but you can grab your cell phone, which I'm sure most of us always keep within arm reach, and you'll see that it works quite well and has a format and outline that lends itself to that. Almost done with the slides here, or at least in this section, but I wanted to highlight some things again so that you're thinking about these. Some you can see, some you can are less obvious but are there. You're going to see that we've moved to this responsive single page navigation style. There's a bunch of accessibility features, and we're working with consultants to help us do a better job there. I'll spend time on Oxford Nanopore, which is a really exciting technology for the classroom. And of course, you won't see it, but it's there, the secure and resilient backend, and some improved tools and features for data management. So I'll highlight those during the demo. As I mentioned before, we are specifically focusing on Blue Line. There will be additional webinars as we introduce new lines and functionality, so you'll get a chance to see that. Um, so Blue Line, just as a refresher, is the line that specifically focuses on DNA barcoding and sequence search analysis. So you're going to be working with a species identification for DNA barcoding, but there are other things that are not quite DNA barcoding, but that operate with a similar workflow for analysis. For example, if you were analyzing mitochondrial sequences, it would be very similar. But you're going to see us go through all the major steps live, and we'll get a bearing for that. Um, just giving a little bit of context, but then I'm going to have another poll question to help me guide through the live presentation. I just wanted to emphasize a couple things about the design, and then that also is the structure that I'm going to present it. Number one, the blue line now has two separate modes, if you will, of working. One of them is based around Sanger sequence, which is what you are familiar with if you use the 1.0 version, where you start with some Sanger sequence. Then there's a select step where you are working with those sequence data and then going to trim, pair, generate a consensus file or consensus sequence, and then that gets analyzed with a set of tools like BLAST or Muscle or Phylop. Um, on the right-hand side are all new features because that mode of nanopore sequencer is totally unsupported for the previous version. So in this case, I'll mention base calling for those people familiar with it. I'll go into depth there. But in that case, you're going to be working with nanopore sequence data, uploading it, and organizing it, and then filtering it, trimming it, and generate a consensus. And then you pour back into the same set of analysis tools, BLAST, Muscle, Phylop. So at that point, it becomes entirely familiar if you're used to the original workflow. Okay, so at this point, I'm going to stop sharing my screen and transition to sharing the actual live workflow. Let me take a moment to quickly comment on the questions that are outstanding before I do that. Um, One was asked about the recording. That'll be sent to everybody, so don't worry if you registered, even if you didn't attend, we'll go ahead and do that and send that after the webinar. I'm not sure what the other question about registrances are, so you're going to have to um, ask that question again. And the only outstanding question about other lines I will get to in a little bit. Okay, so now what I'm going to try to do is move us over to sharing my screen with the new workflow. All right, so great. And I am going to walk through and I am going to try to look at my clock to make sure we stay on time. And like I said, apologies in advance. I'm going to go through this at a fairly decent clip. And as I go through this, of course, you can dump into the Q&A with your questions there, but then also feel free when we're completely done with the demo, I'll reserve a little extra time at the end. DNA Subway still has a feature where you can use it as a guest that requires no registration. Oftentimes, that means there are some limitations to what you'll be able to do, but I just wanted to let you know that if you're using this and you quickly need it to work in the classroom, account registration is not required, but it is desirable. If you don't have an account, you can go ahead and create an account. And now is a great time to mention that if you have a DNA Subway 1.0 account, that will not port over to the new DNA Subway. So you're going to have to create a new account. This is also, and by the way, one of the reasons why we're doing these webinars is to help people transition from 1.0 to 2.0. It's also a good time to remind you that data sets and projects that you might have on DNA Subway 1.0 with Cyverse will not automatically be moved to DNA Subway 2.0. So if you have data sets or experiments or things there that you want to use in 2.0, you want to download or export those things. I will say that sequence data that you've sequenced, for example, with GeneWiz and Azenta, we still have access to those. So there will be more instructions, but at some point you will not be able to access 1.0. It's still up, still exists, but that is not guaranteed. So if you were creating an account, you would create one here. But I just wanted to show you the screen. I am going to go ahead and log into my existing account. And when you log in, what you will be seeing is that um, very similar to what you've had previously, you have a listing of projects. At this point, I already got questions that I generated for myself. So let me quickly say that these data and everything here is no longer stored on Cyverse. It's stored partially on Cold Spring Harbor resources, partially, well, I would say mirrored on Jetstream, NSF Access. So these no longer rely on Cyverse. So I've answered that question. And the next question I've gotten, I'll address a little bit later about loading Nanopore FASTQ files. That's a little bit ahead of where I am right now. But for everybody else, you will have, as you create projects, some projects that are there. If there are any public projects, those public projects will be also visible from this dashboard. And I'll also note that you have the option to delete projects. You have the option to click the info button and edit the name of your project, change it to public, etc. So many of the things we hope you'll be intuitive, but of course you'll see them, and then we have help, and I'll mention that towards the end of my presentation. For right now, I'm going to head and say create project and go to blue line. And what I'm going to do is I'm going to first walk through an entire project using Sanger sequence, and then I'll come back and walk through another project, and this time I'll be using Nanopore. But I'm going to call this webinar February 24. And you can give a description, which is optional, but I'm going to go ahead and say create project. Okay. When I create a project, you'll notice again, everything is one page and we're sort of moving from top to bottom. And there are a number of things that you can do. The very first thing is you're going to want to import some sequences. I'm going to actually click more options, and you'll notice that there's a whole bunch of options. Just about all of them, except for one, are familiar to you if you've used the original DNA Subway. The first one, which is important to me right now, is I'm going to go ahead and click on sample sets, and you'll see that there are a variety of sample DNA sequences that one could use. I'm going to go ahead and click on the Cold Spring Harbor 2010, which are some plant samples, and I want to add these to my project. So I get a little check mark that I've added them, and they're going to appear down in my list of samples that have been added to the project. I've actually made the mistake. Well, I could say it's a mistake. I could say it's educational. I've added them twice, so you could see, because I clicked twice, but I'll get to that in a moment. Okay. The other thing that you can do is you can add trace files. And so just as you may be familiar with the previous Subway, you have the ability here to add files if you followed our protocol for sequencing with GeneWiz Azenta. then those files will automatically be added here. I'll click on RE's files. I don't know what these are, but if I look, I could see these are M13 sequenced of. I have to sort them before I know for sure, but I have a guess. And one could click to add those, and those that works very much like what you would expect. When you are in this interface, you also have the ability to search. So you could put in a name, or you could click in and see by the trace file number, or rather the tracking number. You could sort by mitochondrial, ribosomal, etc. I'm not going to do that now. It's probably taking a few seconds to go in, but just get the idea that you have these cool features to import those sequences. I'm not going to try every single possible method of import, but you will just know that those are there. There is one thing that I do want to highlight because it is um, this is a definite new thing, which is called sequence collections. So with a sequence collections, this is a highly requested feature of you to be able to gather sequences and name them into a collection. And then with those sequence collections, you could decide to search by username or by email, and you could actually publish sequences or gather sequences together and share them. So in my case, I don't remember that I've actually created one that's public here, so I won't be able to see my own. But what you'd be able to do is create. These are some public sequences that are in a collection in my case, but you could search for sequences or share them. And what this allows you to do as an educator is if you have a bunch of sequences that you want all of your students to access, you can distribute them by creating a collection. As you have sequences down here, which is what I'm about to move to, you would have the ability to say, save these to a collection. So it's a really handy feature, and let us know how it's working for you. The other sequences, of course, they're explained in help, but I think most of them are quite self-explanatory. Okay, so that said, um, as I'm adding sequences to my project, you could see right now that I've added sample sequences. If I wanted to, for the sake of just having one example here, I could choose COI sequences, and I'm not going to guess at what these are from this person, but I could add them to a project and they would appear down here, right, as imported sequences. So things start to get grouped. I will not use those sequences since I don't know the person or have permissions, just for sake of showing. And for any given sequence, I can, one of the first things I might want to do is view the sequence. So when I click on that, I'm actually going to be presented with the sequence itself. I could copy to the clipboard, I could download a FASTA file, but one important thing that I probably want to do is I want to toggle because these sequences are all associated with trace files. They're Sanger sequences. So I clicked on the ABI view, and now I'm viewing the trace file, which I can scroll. And of course, I can alter the X and Y zoom to have a clearer look at the sequence quality, see where there may be uncertainty, and also start to see where the areas where the quality starts to um, peter out. So this is fairly familiar, and anything that is associated with a trace file, you'd be able at this point to view. Um, I'm going to move through to the next sections, but just keep in your head that anytime you're adding files to your project, and you can add files to your project at any time, they're always going to be in this section called project files under the select. So this is essentially the space where you are managing your sequence data. Let me keep going because we still have a lot to get to. So I can either click on edit or go to next stop. And now I have three functionalities that I usually am going to do. The first one, the trimming sequence, is where I want to trim the 5 and 3 prime ends. Is go back quickly. But as I think most people will be aware, let me look actually at the sample. Most sequence data, I'll click on a different example. The 5 prime and the 3 prime end are going to have these ends. So the sort of unreliable reads towards the beginning of the end. And usually for the sake of analysis, you want to do some trimming. So back here at the edit step, you could choose those files. I'm going to scroll down to my sample sequences, and I can click on any individual sequence, and I can manually decide where to trim. And I can do that by clicking on any given nucleotide, which is going to create a line and mark the point at which that trim should begin. As you may also notice in the lower left here, I am getting a trace file so that I have the opportunity to see um, this uh, this sequence at that location. I've got a question that I do want to pause to answer now because it's relevant. We're looking at it. And one of those questions is, what is a trace file? I don't have time this second to go to the animation that I'd like to show, so maybe I'll pull it. But essentially, the trace file is the raw data that a Sanger sequencing machine, at least the modern ones that we use, which use fluorescence, that trace file is showing the intensity of the read, the data that it came off the machine. So the intensity, each one of these nucleotides is fluorescently labeled, and the greater the peak and the sharper the peak, the more certain that we have a really good base call, that we really understand that that T is a T or that A is an A, etc. However, we do know that that sequence quality, just by a matter of chemistry and electrophoresis, is a little bit less reliable at the beginning and the end of the sequence. So if I was doing this, I would trim the beginning and the end to get rid of any of the lowercase n's, and then also any that lowercase g that's right here, the lowercase is also a signal that it is probably a lowercase nucleotide, or rather a lower quality. Now, if I click Save Trim, it would save the trim. I could also, this one is done, so I'll pick one more quickly and then I've got to keep going. Let's pick out the reverse there. I can also click on View Suggested, and then the computer is going to give its suggestion based on ends. You could be more aggressive or less aggressive. The option, though, you're more likely to want to do if you have a lot of sequences is to click on Auto Trim. And Auto Trim is just going to take the computer's best guess at what should be trimmed. So it does that. And if you click on or go to look at any of those trace files now, you're going to see that the ends have been removed. Okay. The next thing that we want to do is pair sequences. So we make recommendations when you're sequencing that you follow a naming convention, because here it's quite clear that student 13 plant A R and student 13 plant AF, uh, the F standing for forward and R reverse, that they are actually two reads of the same sequence. So they're the exact same PCR amplification, but they belong together. So you can click on the auto pair, which is what I just did, or you can manually indicate that some sequences are paired. Um, and then you would have the ability. So if I go here and undo the pair, just to show you what that would look like. Now I need to go flip through a little bit. Um, if I wanted to, I could highlight those two sequences, or I could go and indicate that this is a forward and this is a reverse, and then it would give me the option to say, do I want to pair those, right? I'm going to go back to auto pairing just to finish this. And of course, if something couldn't be paired because maybe my naming is wrong, that would be the case to manually do that, right? This is a forward and this is a reverse. So I could say that this is redo for, but now I drop the forward and reverse because what I'm going to create is labeling the pair. In this case, these have some errors, so it's not letting me pair, but I understand that. I understand why. All right, I've got to keep moving, so I'm going to go ahead and edit the consensus sequence. And let me just quickly see of the questions that are here. One question just before I move on. Yes, lowercase letters are poor base calls. So I've answered that. Okay. Another question is, would you recommend running forward and reverse for sequencing? Could you just run one of them? The question has disappeared while I was going, but yes, you don't necessarily have to run a forward and reverse sequence. It's not required. The only case where we would say we really recommend doing a forward and reverse sequence is if you intend to submit the sequence to GenBank, which is one of the features that we do have, because GenBank will require that you do them both. All right, let me keep moving along. So in my edit consensus, I've generated a whole bunch of pairs. So you notice that the naming no longer has F or R. But now the next question is, um, when I add when I've taken the two sequences side by side, as a matter of the sequencing chemistry, those places where the sequence, the forward read and the reverse read overlap are not going to be exactly matching up. One sequence may have started a few base pairs upstream or downstream, and so there are going to be these gaps, these little dashes where they don't you don't have two reads generating a consensus, which means that really, even though I have sequence data here, it's not as high, it's not verified by second read. So what I'm going to do here is trimming, just as it did before. But if you look, now it's pulling the forward and reverse trace file. And you could see that in one read, the forward read, the trace file quality, that little blue bar, which I'm going to try to zoom in, that trace file quality that generated that T is it's a low quality read. Here, it's a high quality. Now, I guess it's nice that they both said T, because that's the best answer. But here, it's more reliable because we really have a better read. So the object of what you would do here is do some aggressive trimming on both the 5 prime and the 3 prime end to make sure that you had the best quality sequence. And then you could save the trim. And now we have a consensus sequence that is high quality. You can go through and do this for all. I'm afraid I don't have time to do this for all. But you would be able to go through and make those or edit those consensus sequences. You can make them and then edit them if you feel you need to redo them. Okay, I do see the questions in the Q&A. I will get to them. So they're not any questions that are exactly the point where I'm right now. So I'm going to go ahead to the next stop. All right, so now at this point, um, we've done all that editing and trimming, and now I may want to do a blast search. And if you're doing barcoding, this is exciting because, well, it's always exciting to do blast search, we know that. But this is even more exciting because it's often at this point that you have a tentative species identification. So I'm going to go ahead and select a few plants. These all happen to be plants. I know what they are. And I'll go ahead and click blast. And as I click blast, it'll take a few moments to go through the workflow, but those blast searches will start running here. When a search is complete, which does take a couple seconds, you'll get a box that allows you to view the results. And when I click on that, I'm able to see those results in a format that it should be familiar to people. We have a bit score, e-score. Of course, I think one of the more important value adds is that I can go ahead and click on a result and potentially pull images from Encyclopedia Life or Wikipedia search. So that's great. If I click on a different one, and different images. Not guaranteed the quality of them. You may need to click there to get high quality images. I do have a question that is relevant, which is an old DNA subway, it was not live NCBI blast. What is it for new DNA subway? And the correct answer is no. We still do not blast on live NCBI servers. We do regular updates so that we kind of keep our blast database up to date with NCBI's current blast database, but it's a local blast search, so it's not being done remotely. It would take forever and it would get slower and slower and slower to the point where we get blacklisted because we'd have so many students blasting at once. Might not work that well. I want to point out that as you do your blast searches, you can also select those blast searches, as I have some checkboxes on the left of my screen here, and then you can click Add to Project, and we can use those in a little bit to be able to do some comparisons with. All right, I am going to keep moving because we have a lot to get to. So at this point, I am going to be able to do a multiple sequence alignment. But first here, I might want to add some reference data. So I do have some databases. I know that I was looking at COI, so I could sort, but I know that, I'm sorry, RBCL, so I could sort. I know that when I'm primarily looking at our plants, I'm going to go ahead and add a reference data set here. And note that when you add that reference data is back available in your sequence data. So remember, keep thinking of your project files as always being visible back up here where you can see blast hits, reference data, your consensus, all of those things that you've generated, those are here. It's a good time now also to say again, not only can you view those sequences, you can rename them, you can save them to a collection. When you click to view them, you can also download them. Keep in mind, for example, this is a blast search, so there's no more AB1 file because it wasn't saying your sequence, but I could still download the FASTA file. So we really try to make things that's as flexible as can. All right, let me go back to the last things I need to do, the muscle alignment, and then also the, okay. Now at this point, I need to choose some sequences to align. So I'll choose a couple of my blast hits. I'll choose my plants. Now notice that I haven't gone through these plants and trimmed the consensus for each one of them. So, you know, there's some ways I could have improved this. I'm going to also add some of my reference data. So for some reason, I am just going to go wild here. These reference data were picked to be like really common species, but also major representatives. All right, so I am preparing myself for a bit of an overload here, but I've gone ahead and I've clicked on muscle. It gives me a chance to talk and I'll answer. Well, I was going to answer some questions, but it happened really quickly that we did this muscle alignment. All right, so in the muscle alignment, we have a whole bunch of clades here, and you can already see some patterns. It's clear where my blast searches are because they have these really common patterns. It's kind of the barcode UPC flavor here. But when I zoom in, really what we're seeing is that each one of those tick marks represents a polymorphism between the overall consensus. If you were to sum the average of all the RBCL residues, and you could see where there are common polymorphisms reflected in the actual annotation here, but also in the variety here, the sequence variation. I'm going to zoom back out, and you'll also notice that not all of these sequences are the same length. So I'm going to want to do the auto trim because when I make a phylogenetic tree, I do need to be looking at least at the same section of the RBCL gene. I'm not going to dig into it here, but there is a similarity table that looks at and allows me to do a pairwise similarity comparison between all the sequences. But what I'm going to do here is I'm going to quickly make some phylogenetic trees. I'll choose a fern, since that is um, out there in more ways than one. And I have the ability now to go ahead and generate some trees. In this case, I've generated a maximum likelihood tree with the fern being the outgroup, just yeah, to expected, right? We have bryophytes, we have the gymnosperms, and then we have all the angiosperms, all of those here, Poaceae. The reference data in this case are nicely labeled with the family names. I don't have time to go into this at a detailed, but there's some things we'd expect to see. So, for example, we knew that plant A looked like it was a holly, and in fact, we have that cluster group that exists here, right? Um, so you could also add distances to this if it makes sense, depends on your interaction. And this interactive tree allows you to move this around and manipulate it. You can download this file as an image or as a string for analysis in different software, or there is also an export feature for some additional manipulations using a third-party software tool called TidyTree. Let me quickly answer one or two questions that have come up and before I move on, and I really then need to keep going. So for Heather's question, these trees in themselves do not, this has distances. if we go and generate, let me just click and do the same thing here. The question was about bootstrapping. And so in the case when you, bootstrap doesn't really apply for the ML method here, but here you can do a neighbor joining tree and you can get some bootstrapping values for those. So I've answered that. I think I've answered about ants exporting the tree, so that's good. All right. Fiona asks a great question, which I'll get to, but we do have documentation that will help you if you're using the Carolina kit. So we have documentation so you can continue your analysis with Subway 2.0. And let's see if I could take one more question. Um, Ida mentions special internet connection. I am on my home internet connection, which I'm afraid of sometimes. So hopefully it will be quite quick, but we could talk more about if there are internet connection issues that we could do to if you have some issues with speed, but that was relatively fast. Okay, if you thought that this was fast, if you thought my analysis is fast, I have taken a lot of time here. So I am going to go ahead and create a new project. But if you have questions about Blue Line that I haven't covered so far, dump them into the chat because once I'm done in another 7 or so minutes, we'll have time for as many of those questions as I can get to. Of course, that'll be over the 45 minutes, but you do understand. So let me create a Blue Line project. And for the Blue Line project with the Nanopore mode, um, what I am going to do is I'm not going to do the analysis with the BLAST part because that is the same BLAST and multiple sequence alignment and trees. It works the same, but there are definitely features on the Nanopore line. So just to quickly, very, very quickly mention, although I'm not going to do it here, I'm going to come back to this. One of the things that DNA Subway has, or rather the DNA Learning Center has been working with um, is these handheld nanopore sequencing devices, which some of you may be familiar with, but some of you maybe not. And so this nanopore sequencing device is a handheld sequencer that we have a whole bunch of resources for working in the classroom. I wish I could talk a lot more about that, but for suffice it to say, one way of doing DNA sequencing for barcoding and for other reasons is instead of sending the sequence data away, you will be able to sequence it in your classroom if you're set up with Nanopore, which is reasonably, relatively affordable. And a major thing, I'm going to go to the dashboard just to highlight, is that a very first part that you might be stopped with nanopore sequencing is that it does require a little bit of specialized compute to transform the raw nanopore data into FASTQ files, into sequence files. So if you are sequencing with Nanopore, a new feature that we've given you on DNA Subway is the ability to do base calling. That's the computational part, usually requires GPUs to transform your sequence data. So this is by permission. You need to go to the dashboard and verify that you're an educator, verify your account. But once you do, you'd have at least limited access to upload your POD5 files, choose the base calling model and the sequence type, and run base calling. It can take hours, but once is done, depending on how much data you have, we will have limits, so we're still finding those out ourselves. But once you do, you'd be able to have those data available to you in your Nanopore project. Let me go back to the project because I do have to get through this. So I can go to some sample data, and in this case, I'm going to choose some COI samples from a data set that we generated in the summer. These are various insects um, that are used forensically. In other words, I believe in the forensics setting, these are insects that you might find near a dead body and you might identify it. Notice that here I can, well, in ordinary terms, rename the sequences and save them in my collection. I can't do that with sample sequences, but I just wanted to emphasize that at this step, there's no view sequence button because there's not really a visualization of raw raw data, FASTQ data, at least not one that we presented you with. So once you have the files that you're interested in, you could also upload them from your device. But once you have those, you're going to move right to the edit step, and then you are going to be presented with a number of options. I'm just going to do the first few of them just so you see that they're what they are. But it's in some ways the equivalent of what you're trying to do with the Sanger reads. We have three tools where we have a little bit of an explanation and documentation links to. The first one, FastP, is a tool that allows you to filter the reads by their quality and get some overall statistics about your job. The second tool, PoreChop, will allow you to remove sequencing adapters. And the final one, Medaka, is where you build a consensus sequence. So I'm running some of these. There really, at this point, is not a lot that you can do except run the tools. We will have some features in some cases where there may be some options that you can exercise with these tools, but they are fairly straightforward. What's not, I don't want to say straightforward, but what is bears digging into is a more thorough analysis than I'm going to give in this particular analysis of what all of this data mean. But in this case, we had a collection of data, and that data in this case has 427 independent nanopore reads. And then after some light filtering for quality, 320 reads survive that, and their average size is about 460 base pairs. Um, there are some this log scale, looking at the distribution of bases, um, hexamers, etc. These generally aren't too important except when we start to get to real long read lengths, but they're there, and it's some information about the overall statistics of your sequence data. The next one, PoreChop, just gives you a report and helps you understand which sequencing adapters might have been detected in your data set. In this case, we were using barcodes. Um, so nanopore data can be barcoded and multiplex, and it's just indicating to you that, hey, it found some adapters. You don't want to analyze them because they're not really part of your data. You want to just get rid of them. And finally, this tool Medaka, which is a really informative one. Here we are viewing the result of the Medaka tool in the integrated genome viewer. What we're actually looking at is all of the individual nanopore reads lined up, and then with each one of those reads, as I zoom in here, you can see that nanopore reads inherently have a higher error rate than Sanger reads. And so there are going to be gaps where a read ends because it's a different size, or there's going to be polymorphisms, which are usually sequence errors, or there may even be indels, deletions, and insertions. But when you average all of those things together, you essentially get a really high quality read because you can subtract those errors, which are largely random. So the whole point of working with at least for barcodes and nanopore data, the other ways we're going to support nanopore data, which I'm going to get to at the conclusion, you would be able to generate a consensus sequence, which I've done here only for two of those reads. But after you have your consensus, you can view that consensus and download it as a FASTA file, and you would proceed to go to the analysis where you would have a blast search. And I'll just do one. I've already forgotten if it's the, well, they should be equally good. I only looked at one, but they should be equally good. But I can click to do a blast search, um, and it will take a few seconds. But basically, from there on out, you might as well have been working with Sanger data because you essentially are blasting that read, generating a multiple sequence alignment, potentially with reference data, and then you are going to generate trees. So I'll leave that to run. I forgot what number four is, but we'll come back to it. Let me finish out my slides and then go into questions because I'm sure there's a lot. And with the 10 minutes that we have, we can just about cover everything. So going back to slides, let me cover a couple of quick things that I want to make sure I share, and it will answer at least a few questions that are outstanding, and I'll return to the rest. So, resources. One, I wanted to just point out that the resources for the barcoding, since I focused today on Blue Line and particularly in barcoding as the application, please, if you haven't done it in a while, visit DNA Barcoding 101. That's one of our sites where we have all of those protocols in depth. There are also explanations there for nanopore data. On the right-hand side is Nanopore for EDU, which is an ebook which goes way into depth on nanopore sequencing, so a little bit deeper with guides, tutorials, and protocols for doing nanopore sequencing, including beyond just barcoding. Of course, go to dnasubway.org/help. Right now, that help page focuses on the two modes of Blue Line, and as we release the other lines, that page will also grow with them so that you're going to see there's documentation for everything. Question that I've already gotten a couple times, Purple Line. So for those who are familiar with DNA Subway, one of the other features, there are others that I'll talk about, but one of the other features, the next one that will be released, is for metabarcoding. So if you are looking at microbial sequence diversity, we had a version, well, we had the original 1.0 Purple Line, which allows you to analyze those data using our version or our implementation of CHIME, which is an extensive pipeline for analyzing microbial sequence data and looking at diversity analysis. So currently, it's in alpha testing. It fully replicates the Illumina pipeline. And what we are working is to release the beta version, which will then be available for people to use and give feedback on. And we're also working on a new version improved with support for nanopore sequencing. So we think that that will be ready for beta in March, so next month, which is just a couple days away. And that brings me to the this fuller timeline. So I already mentioned Purple Line beta in March, and then a nanopore enabled version we're targeting for April, and you can anticipate another webinar which will cover those features. Other things, although this is sort of in time order, but I do want to make you aware of it. We are extensively using external evaluation, including user surveys and focus groups, and some of you may be invited to participate. So please, if you do see a query from us, that's one of the ways that you can help us out in our development work. We're also planning for one more line that is targeted for the summer, and that will be for small genome assembly and annotation with nanopore reads. So that would be the ability to sequence a phage, a mitochondria, a chloroplast, small genomes, and then do full annotation using nanopore sequencing, which would be a great variety of experimental applications for classroom use. The other thing that you're going to see towards the summer is that we have a group, which I'll give credit to in just a moment, that is helping us to develop curriculum materials, learning materials, and so you're going to see more of that starting in the late spring 2026. Let me get some acknowledgments out there, and then I'm going to ask you some questions and answer some questions. In terms of acknowledgments, I want to give first a thanks to the advisory board that is helping guide the project. We have members from faculty and industry whose names are on the screen who are helping us to stay in tune with developing Subway according to what is needed most in the classroom, particularly with a view to community colleges. Since I'm here on this screen, and the first sign of the NSF acknowledgement, the U.S. National Science Foundation is the one that supported this work, so we want to give credit to NSF. And then I also want to particularly thank our collaborators and co-PIs, Monica Trujillo at Queensborough and Metal Dahan at Texas Advanced Computing Center, as well as our consultants that have been working with us as direct collaborators, and of course the project staff who makes this all work. Dave Miklos, who founded the DNA Learning Center, envisioned the original DNA Subway and all the accoutrements, including barcoding. My colleagues Anna and Sue and Daniel, as along with Skyla, who really put the platform together.